So in the last two chapters, we covered antigen presentation to T cells, we covered activation of T cells, and now finally we're going to get into the two effector subsets of T cells, CD4 plus and CD8 plus cells, which covers two little mini chapters in the book, chapters 10 and 11. That'll round out nicely our T cell subchapters, and then we're going to get into humoral immunity with B cells and the effector function of antibodies. So before we jump into the CD4 plus chapter, I want to give a quick recap, an overview of T cells and some of the difference between humoral immunity and cell-mediated immunity. So the defense against microbes that is mediated by T cells is what we call cell-mediated immunity because these cells are directly involved, in contrast to what we term humoral immunity, in which we think of B cells and antibodies. Because remember, humoral immunity is not mediated by B cells. B cells are sort of indirectly involved in that they're simply factories that produce what is the mediator of humoral immunity, which is antibodies. So think of antibodies as carrying out the effector functions of humoral antibody, and then T cells directly, which is why we call it cell-mediated immunity of that particular subset of immunity. In chapter 1, when we talked about adoptive immunity, not to confuse that with adaptive immunity, but adoptive immunity can be transferred from an immunized donor to a naive host via antibodies and humoral immunity. Okay, that's when we think of serum treatments that have just antibodies. You can transfer that to a naive host and impart humoral adoptive immunity. In order to do that cell-mediated immunity, you need to transfer T lymphocytes directly from an immunized donor to a naive host. One of the big key differences between the two branches of immunity is that antibodies can neutralize and eliminate extracellular microbes and toxins accessible to the antibodies. Those antibodies, however, can't reach the inside of cells. So if you had a cell that's infected with a virus or a bacteria that's been phagocytosed but has escaped to the cytosol, antibodies are completely useless. Okay, so that's where cell-mediated immunity comes in because they provide defense against intracellular pathogens. So depending on the location of the pathogen, that's where these two branches of adaptive immunity split. Okay, antibodies are great at enhancing phagocytosis of extracellular microbes, but remember, they can attach to microbes that live or survive inside of cells or that have attempted to be phagocytosed but has escaped to the cytosol within the cell. So again, that's where cell-mediated immunity comes in. That's where T cells come in. They're provide defense against intracellular pathogens, and they're split up into two major subclasses. One is the CD4 plus and CD8 plus cells, which we're already familiar with. The CD8 plus cells are effector cells capable of killing cancerous cells and microbes that are inside the cytosol region. So we want to think of viruses because they typically take over DNA replication pathways within a cytosol of cells. CD4 plus cells, on the other hand, tend to mediate their functions through cytokines. So don't think of them as directly killing cells like CD8 plus cells by directly linking up to other cells. They might link up to other cells, but then they use cytokines to help mediate their functions and instruct other cells what to do. So they serve a critical role in phagocytosis-mediated elimination of microbes by enhancing the phagocytosis process through cell-mediated pathways, as opposed to like antibodies that can do the same thing by enhancing phagocytosis through humoral immunity pathways. Now, in our course, we skipped over the chapters that discussed gene recombinations and the development of lymphocytes. So I first want to introduce a video that gives a very brief overview of that. I'm going to talk a little bit about the development of these cells, just so we know what the origins of CD4 and CD8 plus cells are before we talk about their effector functions and the history of their discovery.
Lymphocytes are selected at multiple steps during their maturation to preserve only cells with useful specificities. Selection is based on the expression of intact antigen receptor components and what they recognize. Many attempts to generate antigen receptors fail because of errors during the gene recombination process. Therefore, checkpoints are needed at which only cells with intact functional antigen receptors are selected to survive and proliferate. Pre-lymphocytes and immature lymphocytes that fail to express antigen receptors die by apoptosis because they do not receive necessary survival signals. At the end of the process, the cells undergo positive and negative selection. Cells that express functional receptors that weakly recognize self-antigens will survive. This process is called positive selection. Strongly self-reactive B or T cells are eliminated to prevent the development of autoimmune responses. This process is called negative selection.
So that video provided a nice little overview of the development of these lymphocytes. I'm going to talk a little bit more about their development here, just because I think it's important to know where the naive version of these cells come from. So first thing I want to point out is that the naive version of CD4 plus and CD8 plus cells are all generated in the thymus. And then from there, they migrate towards the secondary lymphatic organs where they get presented with foreign antigens. But it's here in the thymus where they develop, and it's important to note that they see no foreign antigens here. They only see self-antigens, which are essential for the developmental process. So a precursor to these naive cells is what's known as a double positive cell, which has both CD4 plus and CD8 plus characteristics, which is nice because then it can recognize both types of major histocompatibility complexes. And this is important for a process known as positive selection, in which these cells need to first weakly bind to any self-antigens simply as proof that the T cell receptors, that the major histocompatibility complex recognition system works. Okay, so if those cells that do bind weakly to self-antigens are continually processed along. Those that don't get their survival signaling processes, and they eventually die through apoptosis. The next step is a process called negative selection, where those cells that react strongly to self-antigens also die as well, because the body doesn't want your lymphocytes, your T lymphocytes, strongly binding to self-antigens. That would cause a whole host of autoimmune diseases. And it's actually this process that fails that ends up producing those same autoimmune diseases. So we talk a little bit more about this in detail in later chapters, but I want to give a brief overview on that quick selection process, because it's from here where these cells start differentiating into CD8 plus cells and individual CD4 plus cells, and then continue onwards past the thymus, past the primary lymphatic organs into the secondary lymphatic organs for antigen presentation. There's also a special subset of CD4 plus cells here that don't leave the lymphoid organs. It's a subset called T follicular helper T cells. So again, helper, think that CD4 plus subset, and they're important for B cells to produce higher end antibodies, which we learned about in chapter 12, but I want to point that out here. So now I want to talk about the historical basis of our understanding of the CD4 plus subset. So imagine we have a naive rat that's given a particular injection of this bacteria Listeria monocytogenes that lives on the intracellular compartments of cells. And that's a really important piece of information for this particular experiment. The fact that this bacteria, despite being a bacteria, lives in intracellular compartments in the host. Okay, so this rat is given a lethal dose of that bacteria, and what happens? It dies. Poor guy. In the next experiment, we give the same type of rat a sublethal dose of the same bacteria. And what happens? Because it's a sublethal dose, it lives, it becomes immune, and then when we next give it the same lethal dose as in the first experiment, it lives and is happy because it already has memory of this particular cell, is much more effective at fighting it, despite the increased dose of that particular bacteria. In a follow-up experiment, we take a second naive rat that's given the same lethal dose of the same bacteria as in the first experiment, except this time we transfer serum, which we know has antibodies, from the immune rat previously. And what happens? In this case, the rat dies. So scientists here were a little confused because they knew that they were transferring antibodies, they knew they had this bacteria, they thought the rat would survive, yet it didn't. Poor guy. So as a follow-up experiment, they recreated the same thing. They took a naive rat, they give it a lethal dose of the same bacteria, and except this time from the immune rat, instead of taking the serum, they transfer lymphocytes, the T cells, and give it to the naive rat. And what happens? This time, the formerly naive rat is alive because the lymphocytes were able to fight off the infection. So at this point, scientists understood that, okay, I guess lymphocytes can kill bacteria. So let me test that theory in a follow-up experiment. So in this experiment, they took activated T cells from that immune rat. They put it in a petri dish combined with that same bacteria. And what happened? The bacteria lived. Okay, so in this case, the T cells did not kill the bacteria, yet they knew that they were somehow involved in killing the bacteria in the rat. So what was going on here? So in another follow-up experiment to confirm to see what had happened, scientists took the same exact experiment, except this time added phagocytes. And what happened? In this case, the bacteria died. So what scientists could conclude from this is that yes, T cells were necessary to kill the bacteria, but they could not kill them directly. Only when phagocytes were added to the dish could they then figure out that the bacteria would die, not because of the T cells, or at least not directly because of the T cells, but indirectly through the phagocytes, which they would later confirm were being told to kill the bacteria by the T cells. Now, in light of those experiments and then some of the things that we learned about in previous chapters, we can say the CD4 plus cells recognize phagocytosed microbes that process their antigens onto class II MHC complexes, but it's the phagocytes that actually destroy the pathogens. Now, from our innate immunity chapter, we know that phagocytes are capable of eliminating pathogens, but here it's where T cells can greatly enhance this. This is also important in that some microbes can resist just the innate mechanisms of phagocytosis and can survive and replicate inside macrophages and inside phagosomes. And it's only until the T cells come in to help, the CD4 plus cells, that they can eliminate those. Now, despite the fact that scientists understood the link between T cells and intercellular pathogens, they could still appreciate and recognize that even within that general description of T lymphocytes and intercellular bacteria, there's a wide variety and diverse array of responses that are included within that subset. So for instance, Mycobacterium tuberculosis, the response to that is dominated by macrophages, yet helminthic parasites that are like large tapeworms are involved in the production of IgE and activated by eosinophils. Then there's autoimmune diseases that are involved in tissue damage by inflammation and accumulation of both neutrophils and macrophages. And then there are other allergic disorders produced by lesions that are marked by large amounts of eosinophils and, say, mast cells. In all these cases, T cells were somehow involved. So scientists knew that there was this diverse array of responses caused by cells. In fact, even in these cases within CD4 plus cells. And the reason for that is that there's different subtypes of CD4 plus cells. And I'll list them here in the beginning. We're going to talk about them more in depth in later chapters. But there's Th1, Th2, and Th17. And if you're curious like me and you wonder what the story is behind that nomenclature and why it doesn't go 1, 2, 3, I think one group of scientists who were studying these subsets sequentially numbered the first two that they discovered, so Th1, Th2, and then completely different scientists named it after a cytokine that's associated with that particular cell. So Th17 is going to be associated with IL-17, as we're going to understand later. In addition to those sort of three effector cell subtypes, there's also follicular helper cells, which are involved in B cell and antibody production, and also regulatory T cells. So those are both types of CD4 plus cells. Now, I mentioned at the beginning of this chapter that these CD4 plus helper cells mediate their functions not directly through cell-cell interactions, but sort of indirectly through the cytokines that they produce. These cytokines then give instructions to the cells that activate the T cells. And for this reason, it's important to understand the link between these cytokines and these three different subsets. So each of the subsets has a signature cytokine and other cytokines that are characteristic of it. In general, it's for Th1 cells, it's IFN gamma. For Th2, the signature cytokine, I would say, is dominated by IL-4, although IL-5 and IL-13 are present. And then for 17, this is again why the cell gets its name. The cytokine is IL-17, and then also to a lesser extent, IL-22 is involved in that subtype. Now, these cytokines help the T cells and then the other cells that they attach to carry out their effector functions. But it's also important to recognize that these cells can migrate to different areas of the body through different chemokines, because every type of interaction is going to require different migratory patterns of the different subtypes. So there's almost sort of signature chemokines that are associated with these other ones in order to get them into the right places where they're needed. So for Th1, they're involved with CXCR3 and CCR5, which are given out by innate immune responses. So we see high amounts of EMP selectin ligands to help bring them to the sites of inflammation. Th2, we see CCR3, CCR4, and CCR8. So they're typically involved in helminthic infections, mucosal tissues, and allergic responses. And then for Th17, it's CCR6. So these are tissues affected by different types of bacteria and fungi. Now, these signature cytokines produced by these cells act as a positive feedback mechanism to induce further polarization of naive CD4 plus cells into those particular subsets. Furthermore, each one of these cytokines even inhibits the differentiation of naive CD4 plus cells into the other types of subsets. So for instance, once you have IFN gamma in the mix, that induces the differentiation of the Th1 subtype and inhibits the differentiation of the other two subtypes. It's a phenomenon known as polarization. These different subtypes can also be involved in sort of autoimmune diseases and other types of inflammatory diseases associated with their overactivation. So for instance, type 1 or Th1 and Th2 are involved in certain autoimmune diseases that deal with high amounts of inflammation. Th17 deals in allergic reactions. One thing I really want to preface with this subchapter and talking about these different subtypes is that there's really a whole lot of gray area between these different subtypes. So there's other subtypes exist, there's subtypes of subtypes. Many of these subtypes are not really clearly defined. They all produce a variety of cytokines. It's certainly possible that Th1 cells might produce a little IL-4 and IL-17. Some of these have overlapping characteristics. And then furthermore, subtypes can convert into other subtypes, so undifferentiate and then redifferentiate. So after that little disclaimer, we're going to kind of treat these as very black and white subtypes, but I just want to put that on your radar for now. Now, the development and differentiation of these different subtypes all occur in secondary lymphoid organs. At the beginning of this lecture, we talked about the development of these lymphocytes and how naive CD4 plus cells are developed in the primary lymphoid organs, like the thymus, and then travel to the secondary lymphoid organs. It's only in the secondary lymphoid organs where naive CD4 plus cells, which by definition have no subtype, first get activated and then are told, primarily by antigen-presenting cells, into what subtype to differentiate into. Other immune cells like mast cells or natural killer cells present in the lymphoid organs also have a hand in driving this differentiation by the cytokines that they produce. There's also other stimuli other than the cytokines that can influence what subtype those CD4 plus cells differentiate into. It could be antigen affinity or antigen amount that help drive it into various different subtypes. So not only are there cytokines that drive the initial differentiation, but those same CD4 plus subsets also produce their cytokines that promote their own development and again suppress other subsets, which I refer to as polarization. For instance, IFN gamma inhibits both Th2 and Th17 production, and similarly, IL-4, expressed by the Th2 subtype, also promotes further differentiation of other naive CD4 plus cells into the Th2 subtype. Part of the main reason why this is important, it's for very similar reasons why CD4 plus and CD8 plus cells have different roles, and thus antigen-presenting cells process antigens in different ways to promote either one of their activation. It's the same thing in the subtypes of CD4 plus cells, as different subtypes of CD4 cells have different effector functions. So Th1 subtypes are useful for intracellular microbes. The Th2 subtype is used in helminthic and very large parasites. And the Th17 is used in certain types of bacteria and primarily fungal infections. Now that we've covered an overview of the different subtypes of CD4 plus cells, we're going to talk about them in detail. And the first one we're going to get to is the Th1 subset, whose principal function is to activate macrophages to ingest and destroy microbes. The differentiation of this particular subtype is stimulated by many intracellular bacteria, just like the listeria that we talked about in the beginning of this lecture, mycobacteria, certain parasites that infect dendritic cells and macrophages, and even some viruses and viral proteins that are accompanied by a strong innate immune reaction or administered with strong adjuvants, like in the case of a vaccine. What's common among all these infections are the cytokines they produce. For instance, they all tend to produce high amounts of IL-12, which tends to be the most important stimulator of this particular subset differentiation. They also produce IL-18, which works synergistically with IL-12, and then furthermore, they produce IFN gamma. Now, earlier I said that IFN gamma is one of the signature proteins associated with this, as opposed to IL-12, in part because IFN gamma works as a positive feedback mechanism where this particular subset also produces that cytokine to promote further differentiation of adjacent, naive, or being activated CD4 plus cells. And they also polarize this particular subset, meaning that not only do they promote the differentiation of this subset, but they also hinder the differentiation of the Th2 and Th17 subtype. The other principal reason why this is the signature cytokine of this particular subset is that IFN gamma is the one that carries out or mediates the function of this particular subset. It gives all the instruction to macrophages and the other cells at this axon, whereas IL-12 and 18, along with IFN gamma, are more responsible for creating and differentiating this particular subset. So for that reason, I want to talk about IFN gamma a little bit more in detail. Now, IFN gamma is the principal macrophage-acting cytokine. As we're going to learn, this particular subset is going to be associated with macrophages, and this is the way that they communicate with macrophages, through IFN gamma. IFN gamma itself is a type 2 immune interferon. That's in contrast to the type 1 antiviral IFN cytokines that share the name interferon. But I want to make sure we're not confused. This has nothing to do with viruses. It doesn't interfere with viral replication, which is how I think of these cytokines getting their name. It principally acts on immune effector cells like macrophages. IFN gamma is produced by both subsets, CD4 plus and CD8 plus cells, in response to antigen recognition. And this is also enhanced by the cytokines IL-12 and IL-18. So they help signal these particular cells to produce more IFN gamma to carry out their mediator functions. In the innate side, natural killer cells also produce IFN gamma, which is activated by ligands on stressed or infected cells, and also in response to IL-12. So we can start seeing the synergistic effect between IL-12 and IFN gamma. IFN gamma has a number of functions, all of which are related to phagocytosis and in general classical macrophage activation. So this even promotes the M1 subtype of macrophages, which carries out the phagocytic and microbe killing actions, as opposed to M2, which we'll learn is related to the Th2 subtype. It polarizes Th1 differentiation. I know I've said this many times already, but I just want to stress this here, that this cytokine is what helps that. So not only promotes IFM1 differentiation, but suppresses the Th2 and Th17 subtype. It also promotes the expression of surface molecules needed for antigen presentation and activation, like MHC complexes and B7 co-stimulators. It also promotes B cells to undergo subclass switching to the IgG subtypes, specifically not IgG4 though, so IG1, 2, and 3, and also inhibits the IgE subtypes. The reason why these are important is that the IgG subclasses bind to Fc gamma and the C1q complement receptors that promote phagocytosis. Now, specifically for this B cell function, the source of IFN gamma is actually the other subtype of CD4 plus cells I briefly mentioned in the beginning, the TFH cells that are in the lymphatic system. But it still goes to show how IFN gamma can help promote classical macrophage activation through B cells kind of indirectly, and then producing the right type of antibodies that are necessary for enhancing phagocytosis in macrophages. So now we've talked about the Th1 differentiation, we've talked about T cell activation in previous chapters. Now we're going to talk about the macrophage activation. So now these T cells are acting upon the macrophages to carry out their functions. And so this activation, or the killing mechanism of M1 macrophages, requires two key components. They are the signature cytokine of the subtype, IFN gamma, and then CD4 binding of the T cell CD40 ligand. So one thing I want to point out here is that what's not necessary for macrophage activation is anything related to the T cell receptor or the MHC complex. So in this picture on the right, we have all three of those things occurring here. But the MHC antigen T cell receptor is only necessary to tell the T cell, so in this case the T cell is the one receiving the communication, to carry out its functions. The T cell's functions is to then elicit IFN gamma and then bind the CD40 ligand to attach the CD40 on the macrophage. So when those two things happen on the macrophage, they induce the production of nitric oxide synthase, so that's a killing component, lysosomal enzymes, and phagocyte oxidase, which helps produce reactive oxygen species within the macrophages. Although that's technically more related to neutrophils, whereas macrophages most dominantly use the nitric oxide synthase. The main result of this is to kill the phagocytosed microbe, mainly through nitrogen oxide, but also reactive oxygen species. But there's also the secretion of other types of cytokines by the macrophages, like tumor necrosis factor, TNF, IL-1, other chemokines, and other short-term lipid mediators, the collective action of which is to recruit more leukocytes and enhance the host's ability to destroy infectious organisms. So now we've talked about the Th1 subset, which we know is primarily involved in phagocyte-dependent defenses. The Th2 subset is the exact opposite. It's a major effector T cell population for phagocyte-independent defenses. So I want us to not think about phagocytes when we think about the Th2 subtypes. The major cells that they tend to act on are mast cells and eosinophils. They are induced by helminths, which are parasitic worms, allergens, and microbes in the mucosal tissues like the epithelium. What else helps me link this particular subset with this parasite and the way they act is that I know that helminths are too large to be phagocytosed the same way the bacteria are. And they're also resistant to microbicidal activities of Th1 cells. So I think of the Th2 subtype as not being able to phagocytose. So they help non-phagocytes act on these particular parasites. The cytokine profile that helps the differentiation of this particular subset is not completely defined. It's known that IL-4 is heavily involved, and it's also the signature cytokine because this particular subset also produces that cytokine as sort of a positive feedback mechanism. So just like IFN gamma in the Th1 subset, this cell produces its own signature cytokine that acts on itself and other cells to increase their activity and increase their differentiation. Because of their association with mast cells also help produce IL-4 to work on these particular subtypes that would eventually act on them later on to carry out their functions. Now, because IL-4 is the signature cytokine of this particular subtype, I want to talk about that a little bit more in detail, and also some of the other cytokines that work on it, primarily IL-13. So the primary sources of IL-4 are the fully differentiated Th2 subtypes, activated mast cells, and to a lesser extent, eosinophils. These are the cells that we're going to associate with the Th2 subtype. Furthermore, IL-4 stimulates the B cell class switching to the IgE and IgG4 isotopes, which help act on mast cells and eosinophils. And as you know, this is the exact opposite of IFN gamma. IFN gamma inhibits this particular subtype and then promote the subclass switching to the other IgGs. This is going to be a common trend between the differentiation of the Th2 and the Th1 subtype and the functions of IL-4 and IFN gamma, which are those respective cell subtypes. So for instance, just like IFN promotes the switching of M1 classical activation, IL-4 inhibits M1 classical activation and promotes M2 activation in macrophages. Together with IL-13, they stimulate peristaltic and mucus secretions in the GI tract. And they also help recruit leukocytes like eosinophils by promoting expression of adhesion molecules and chemokines that are specific to those particular cells. IL-13 on itself is primarily produced by the Th2 subtype cells. And what's important here is that that particular subtype does not express IL-13 receptor. So while IL-4 is produced by these T cells and then it acts on those same or adjacent T cells, IL-13 does not have the same type of reciprocal function. Okay, it's produced by these cells, but they can't recognize it themselves. It has no mediated functions on the T cells themselves, only the mast cells and eosinophils. For this reason, we know that IL-13 is not involved in Th2 differentiation or polarization. Now that we talked about the cytokines and differentiation of the subset, let's talk about the defense mechanisms and also some other repair mechanisms that the Th2 subset is involved in. So first of all, the Th2 subset is sort of indirectly involved in eosinophil-mediated interactions due to the cytokines that they secrete. IL-4 and IL-13 stimulate the production of helminth-specific IgE antibodies. Now I say specific in that antibodies in general would be specific to the foreign antigens that are present on the helminths, but it's the Fc portion that are specific to the receptors on eosinophils that causes some of their degranulation and their eosinophil fighting mechanisms, which include the release of major basic proteins called natural killer cell activators and cationic proteins that sort of on the extracellular scene act upon these particular parasites. So in this case, we want to think of the IgE antibodies as opsonins that very efficiently coat the helminths. They can also work independently of coating or opsonization by causing the degranulation of mast cells, which act in very similar fashions to eosinophils. And this is where the implications in allergic functions come into play, because mast cells are big mediators of allergic reactions. I also mentioned earlier that Th2 subset is a big defensive mediator at epithelial and mucosal barriers. That's because they induce mucus secretion through peristalsis, they promote the expulsion of microbes through the mucosal organs, and for that reason the subtype is sometimes referred to as the barrier-mediated defense. In addition to parasitic defense mechanism, the Th2 subset has also important replications in the wound repair and tissue repair process through alternative macrophage activation, where they promote the differentiation of this M2 subtype of macrophages, which promotes collagen synthesis, fibroblast proliferation, and angiogenesis. The end result is that it terminates inflammation and promotes wound healing. The last subtype I want to touch on is the pro-inflammatory T17 subset, whose primary purpose is to recruit neutrophils, and to a lesser extent monocytes, to the site of inflammation. This particular subtype is critical for destroying bacteria and fungi, microbes that are killed by phagocytosis, and also contributes significantly to certain inflammatory diseases. Their differentiation is stimulated by the presence of these bacteria and fungi, which prompt dendritic cells to release certain cytokines at different stages to promote their differentiation. In somewhat of a sequential order, these start off with IL-6 and IL-1 in early stages, and then later in both the later differentiation and maintenance IL-23 and IL-21. What's kind of unique about Th17 is that IL-21 is the same positive feedback mechanism like how we've seen with IFN gamma and Th1 and IL-4 with Th2. However, this certain positive amplification process that both the Th subtype releases and acts on itself is not the signature subtype of this particular subset. That would be IL-17. So whenever we think of the signature cytokine, think of what mediates their effective action, not necessarily what their differentiation is. Although there is a lot of overlap, particularly with the Th1 and 2 subtype, but less so with the Th17 subset. And again, I'll touch on the topic of polarization. The differentiation of Th17 is inhibited by the signature cytokines of Th1 and Th2, which are IFN gamma and IL-4. So any of the differentiation of those other two subsets suppresses the differentiation of the Th17 subset. Because IL-17 is the signature cytokine of this particular subset, let's talk about it in a little bit more detail. We'd already discussed this previously in Chapter 4 because we know it's a prominent cytokine of the innate immune system, and now we're learning how the adaptive immune system, through these T helper cells, can help augment the action of these innate immune cells. And so in this case, it's through this cytokine IL-17. Because IL-17 tends to act on its signature cell, which is neutrophils, it should come to no surprise that IL-17 induces neutrophil-rich inflammation by promoting the expression of chemokines specific to neutrophils, like the CXCL8 ligand that neutrophils like to attach to. IL-17 also enhances neutrophil generation by producing the granulocyte colony-stimulating factor, so this is a growth hormone, GCSF, and also stimulates the production of antimicrobial substances like defensins that are important for the inflammatory process. There are other cytokines involved with IL-17, like IL-22, which is used by this subtype and produced in epithelial tissues to promote the barrier function of epithelia. This also has some overlapping functions with IL-17, such as the production of antimicrobial defensins. And then there's also IL-21, which I previously mentioned was the positive feedback amplification mechanism of these cells. These cells both produce IL-21, which further promotes differentiation of that particular subtype. And it's also used for the generation of follicular helper T cells, which activates B cells in the germinal centers of the lymph nodes. Now to touch on the roles of both this particular cytokine and then the subset Th17 CD4+ cells, I'll again reiterate that the primary purpose of the subtype is to recruit neutrophils to the site of inflammation. Th17 cells are also involved in various roles for maintaining the integrity of epithelial barriers. I won't go into too much detail about these. There's a lot of evidence to suggest that there's subsets of this particular subtype that all carry out these various functions. But in general, what they do is that they stimulate the local antimicrobial peptide productions like defensins, and they also express IL-22, which helps promote regeneration of the epithelium. Furthermore, there's actually a lot of interesting diseases that are involved in both the deficiency and overactivity of this particular subset. One particular genetic deficiency which results in defective THC development is called Jobe Syndrome, named after the biblical figure who was smote with boils by Satan. And these particular patients afflicted with the disease, they have rampant and reoccurring bacterial and fungal infections that make it look just like the biblical figure. Furthermore, the overactivity of this particular subset contributes to a lot of inflammatory diseases like psoriasis, inflammatory bowel disease, rheumatoid arthritis, and multiple sclerosis. So to summarize this chapter, I'll present to you this table, which is actually given earlier in the book, but I think it summarizes things nicely. It shows what the effector cells are and what they're named. It shows their defining cytokines, the principal target cells, and their major immune reactions for both host defense and then also their role in diseases. So remember, always recognize the signature cytokines associated with the effector T cells. Th1 goes with IFN gamma, Th2, IL4, Th17, IL17. We also touched on other types of CD4 subsets like follicular T helper T cells, which are associated with IL21, and B cells. And we'll actually learn about those a lot more in chapter 12, I think, when we talk about B cell production and activation. So in short, I think if you have a good understanding of this table, you also have a good understanding of this particular chapter. And now that we've touched on CD4 plus cells, we can move on to the other major subtype of T lymphocytes, CD8 plus cytotoxic T cells. To give a little background on why these particular cells are important, I'm going to touch on viruses a little bit. So first of all, viruses can't replicate themselves. They have to hijack host cells for protein synthesis machinery in order to replicate. In fact, among biologists, there's been an ongoing debate on what the exact definition of living or being alive. And a lot of biologists maintain that one of the key traits of life is the ability to replicate. And so a lot of biologists argue that viruses aren't themselves alive because they lack this inherent trait. But that's a debate for a different class, so let's move back to the topic at hand. Viruses can't be destroyed if a cell is not a phagocyte with intrinsic lysosomal mechanisms. And furthermore, a lot of viruses or bacteria can escape phagosomes and enter the cytosol anyways, so it could be that phagocyte is in itself incapable of killing a virus. This is what necessitates the need for cytotoxic T lymphocytes, which is also a name for CD8 plus cells. And these are the effector cells that don't kill viruses directly, but they kill the cells harboring viruses. And they share a lot of similar traits and roles to natural killer cells in the innate immune system. We don't want to just associate CD8 plus cells with viruses. First of all, we saw that CD4 cells also have the capability of destroying viruses. But CD8 plus cells are also incredibly important for eradicating tumor or cancerous cells. Right? We understand that all cells can develop cancer, not just phagocytes, so we need these type of cells to kill cancerous cells. And they also have major implications in acute rejection of organ allografts or organ transplants. This latter role has a lot of implications in tissue engineering and regenerative medicine. And we'll be touching on this topic in one of the later chapters towards the end of the semester. Now, I want to touch on the activation and antigen presentation of CD8 plus cells. We learned a lot about this in previous chapters. We learned that CD8 plus cells require antigen presentation to class 1 MHCs. Again, I always think about the T cell subtype and what class of MHCs by thinking that CD8 plus cells in class 1 kind of bookend the numbers. 1 and 8 are the smallest and largest. And then it's class 2 with CD4 plus cells. So they occupy the middle numbers. Furthermore, class 1 MHC antigen complexes are produced by antigens made in the cytosol, not through phagosomes. And we also learned that activation of CD8 plus cells, or really any other T lymphocyte, is best performed by dendritic cells. However, dendritic cells are phagosomes that are capable of killing viruses through phagocytosis. So it's possible that if you introduce this particular virus that's infecting the body to dendritic cells, that the dendritic cells aren't going to allow themselves to be infected. They might just kill them and process them, which would result them being presented on class 2 MHCs, which wouldn't be effective for CD8 plus cells. Furthermore, it's even possible that other cells become cancerous that aren't dendritic cells. So the kind of question is, how can dendritic cells effectively present viral and cancerous antigens to CD8 plus cells if they're not the ones being infected or cancerous? This is where we bring up this cool concept known as cross-presentation, where antigens already present on other cells are processed in whole by dendritic cells, so that dendritic cell might swallow up an infected cell. And even though they bring it into a phagosome, these dendritic cells are kind of smart enough to allow these antigens to escape to the cytosol, even though they're phagocytosed. Thus, being able to process in the cytosol area allows that antigen to be produced on a class 1 MHC, not a class 2 like how phagosomes usually process things. This, in the end, allows dendritic cells to process that antigen to CD8 plus cells and produce an appropriate response by these cells. Now, as we learned in previous chapter, the activation of CD8 plus cells requires co-stimulation via B7 and is best carried out by dendritic cells. Now, activation may also potentially require the help of CD4 plus helper T cells, depending on the situation. If there's a situation that results in a strong innate immune response, which in itself would produce a lot of B7 ligands that would act as co-stimulators, or in the cases where the antigen-presenting cells, like dendritic cells, are directly infected, then the role of CD4 plus cells is not as critical. However, in cases where we have latent viral infections, organ transplants that themselves don't have any infectious agents, or in cancerous tumors, these all produce a very weak innate immune reactions. And this is where CD4 plus cells actually have a really critical role in activating CD8 plus cells. This is perhaps best illustrated by the case in HIV or AIDS. So for all viral infections like HIV, CD8 plus cells are capable of killing the cells with the HIV virus. However, what's really detrimental with HIV and AIDS is that HIV only affects and eliminates CD4 plus subsets of T cells. So CD8 plus cells actually kill the cells that are meant to help activate the CD8 plus cells. And for this reason, it's viral infections that later on, when HIV has progressed to AIDS, that is the ultimate killer of patients with this disease. In fact, AIDS is actually defined by the CD4 plus count. This is what we refer to as the T cell count in HIV patients. And in this case, it's less than 200 cells per microliter of plasma. In addition to co-stimulators and CD4 plus cells, many cytokines are involved in the activation of CD8 plus cells. IL-2 is a big one. We learned that IL-2 can also be considered a T cell growth hormone, as it promotes the proliferation of all types of T cells, CD8 plus cells included. We also learned that IL-2 receptor, the alpha portion, is what is caused to express during the activation process, whereas the beta and gamma chains are constitutively expressed. IL-12 and type 1 interferons. So again, type 1 interferons are the antiviral interferons, and we want to link viral fighting mechanisms with CD8 plus cells. Help stimulate the differentiation of these effector CD8 plus cells, and also to a certain extent the Th1 subtype of CD4 plus cells. IL-15 is necessary for the survival of CD8 plus cells as well. And then IL-21 via the CD4 plus cell subtype promotes differentiation of both effector and memory CD8 plus cells. And since we're also talking about activation and the process of carrying out the effector function of these cells, I also want to bring up the concept of exhaustion, which comes about in chronic viral infections where the activated effector CD8 plus cells gradually stop responding to these cytokines that tell them what to do. I like to think of it as literally these cells being exhausted from carrying out so much work, so then they just stop working at all. And this is in contrast to the concept of tolerance, which is, for instance, when a T cell only recognizes the antigen in the absence of co-stimulators and actually induces a T cell to fail to respond because it starts tolerating that particular antigen. So think of tolerance as before activation and exhaustion as after activation. Certain deficiencies that pop up during exhaustion are decreased proliferation, reduced productions of IF and gamma cytokines, and then in general poor cytotoxic activity. We also learned about the inhibitor functions of certain receptors on these T cells like PD-1 and CTLA-4. We see increased expression of these inhibitory receptors. The exhaustive part of CD8 plus cells is observed in many chronic viral infections, notably HIV and hepatitis C, where we just have these recurrent viral infections. Now, the CTL-mediated killing mechanisms involve specific recognition of target cells and delivery of proteins that induce cell death. So first, let's talk about this recognition complex. First and foremost, what's necessary is the class 1 MHC antigen T cell receptor pairing, right? So this is what gives the T cell its specificity. In addition to that, there's the CD8 co-receptor, which helps stabilize the MHC and the T cell receptor binding. And then lastly, there's adhesion molecules involved in this because the T cells have to get up close to the target cell. So there's a lot of adhesion mechanisms that come into play, but primarily in this recognition complex, it's the LFA, which is leukocyte function associated antigen 1, which binds to its ligand on the infected or target cell, ICAM1. I would like to point out what's absent in this is any co-stimulator like B7 and CD28, because once a CD8 plus cell is fully activated, like through a dendritic cell, it's ready to go at a moment's notice, and it just needs to recognize the antigen in order to carry out its killing mechanisms. These other components, like the CD8 co-receptor and the adhesion molecules, are important for forming what's called the immune synapse. I like to think of this as a conduit through which these cell-killing proteins can be delivered through. So instead of just delivering these cell-killing proteins to the extracellular space in the vicinity of the target cells, this synapse more efficiently delivers those cell death proteins directly to the target cell through this sort of conduit, ensuring that only the target cell receives these proteins and not just other adjacent cells. Once the immune synapse forms, a lethal hit of these cytotoxic proteins that are stored in the T cells are delivered within minutes. Two of the main proteins involved in this actually are identical to the cell killing mechanisms of natural killer cells. The first one is called perforin, which I like to think of as causing perforations in the target cell membrane, which allows the other later proteins to enter and induce the cytotoxic killing mechanisms. So this is similar to the C9 complement protein process, which also disrupts cell membrane proteins. The follow-up proteins, which are now allowed to enter thanks to perforin, are called Granzymes, which are proteases that induce apoptosis by activating certain intracellular capsases within the cell. CTLs also have granule-independent mechanisms for killing cells. The main one is the expression of the FAS ligand, FASL, that targets the appropriately named death receptor expressed on many cells in the body, FAS. So as soon as FASL on the CTLs bind to the FAS receptor on the target cell, that initiates the same apoptosis pathway that Granzymes help induce. After the delivery of these killing proteins, the CTLs release from the target cells well before the cell actually dies. In addition, there are non-contact killing mechanisms that CD8 plus cells are involved in. For instance, they're a big producer of IFN gamma, which induce the macrophage activating phagocytosis similar to that as Th1 cells. In fact, IFN gamma is so closely linked with CD8 plus cells that IFN gamma assays are actually used as an indication of the activity of CD8 plus cells indirectly. Now, because CD8 plus cells don't kill viruses directly, but they kill cells infected by viruses, you can imagine both good and bad consequences of killing host cells. There are good cases, like when cells have mutations that cause cancer, where we do want to eradicate host cells. But there's also bad cases in certain viral infections, like with hepatitis B and C viruses, where the viruses themselves are rather benign. It's the fact that the CD8 plus cells are killing our vital and crucial liver cells that's really the main consequence of the disease. So that's a comprehensive overview of the effector functions of these effector T cells, CD4 plus and CD8 plus cells. Next up, we're going to talk about B cells and the humoral adaptive immune system. Thanks for listening.